Hi folks, so short video today on the life cycle of a virus. So just to get started, before you need to know what a virus is basically. So a virus is some genetic material. So small amount, really simple, small amount of genetic material like DNA basically. And then that is coated in a protein coat. Right. There's our crazy nasty virus. So we have got a protein coat, and the protein coat has a special name. Protein coat is called a capsid, and then inside that you've got the genetic material. So the genetic material in there, and then protein coat. And the protein coat is what helps the virus, helps the genetic material not, that's really fragile, that part. And so the protein coat helps it to survive essentially, so that the DNA of it doesn't break down. Viruses do not have the ability to reproduce, right? They need to use other living things to do that. So that's where the sort of slightly trickier parts of the life cycle come in. So we've got a cell. So we'll say it's a bacterial cell, and then the bacterial cell, I'll do this in green. The bacterial cell has got some DNA in it. So we'll say it's a chromosome. And then our nasty little virus comes along and then gets onto the edge of the cell. Oh, I've forgotten to do the virus's genetic material in there. And then it injects itself into the virus. Sorry, it injects itself into the bacteria cell. So we end up with, this is like the next stage of it. We've got our bacteria cell. It's got its DNA in there, but then it also has got the virus's genetic material in there. And then it sort of hijacks the cell basically. This genetic material hijacks the cell, and it makes the cell make loads more copies of it. So we've still got the, so it makes it make loads more copies of this genetic material of the virus, but it also tricks the cell into synthesizing the protein coats, right? So like the middle bits, and then the bits that look like eggs. So we have produced some of the protein coat, and we've also produced we've also copied the genetic material. Sorry. So virus has come down, injected the genetic material into the cell. It's tricked the cell into making lots more of the genetic material, and it's also tricked the cell into producing the protein coat. This is one of the most common exam questions on this. And to get the marks, you don't just need to know about the genetic material, it's about the protein coat. So the protein coat is produced, and then the protein coat is assembled. Protein coat is assembled. So we've got our bacterial cell. This can happen inside human cells too, right? And we've got lots of these. I'm only going to draw 3, but it would actually be like millions of them. And they've got their, I'm not in red. Where's my orange pen gone? Right. And they've got their genetic material inside too. Okay, I should have drawn this a lot bigger, and then it would have been easier, but hopefully you can see that. And then what happens is run out of space, didn't forward plan. Right, the cell sort of bursts open basically. The cell gets destroyed and all the viruses escape. Need to draw the genetic material in there too. And then all of these viruses can go on and attack a new cell. And so the process starts again. So one virus goes in, injects the genetic material, hijacks the cell, gets the cell to copy its genetic material and also to produce the protein coat. So the protein coat is produced and then the protein coat is assembled. Two most important parts of this for exam questions, right? You need to not just mention the genetic material, protein coat produced, protein coat assembled. Then cell bursts apart and you get millions of these viruses come out and then they all go and attack a new cell. This cycle has a name. It's called, should have used a different pen. It's called the lytic cycle. And this part here where the cell sort of bursting apart, so what we say is the cell lyses and it releases the virus. So lytic cycle, genetic material injected, genetic material copied, protein coat produced, protein coat assembled, cell lyses, releases all the virus, and then they go, right? But it's actually sort of figure of eight, because at this point, something different can happen, right? So at that point, if I stop chucking my pens everywhere, something slightly different can happen, and the virus genetic material inserts itself into the chromosome. So we've got the chromosome there with the DNA from the bacteria, and then the virus kind of just genetic material inserts itself into that. Right, so the virus genetic material has inserted itself into the bacteria chromosome. And then what happens is mitosis. So just like bacteria would normally divide, and mitosis produces two identical cells. So let's just draw this kind of halfway through. So it's in the final stage of mitosis before it's separated out. And it's just copied that, it's copied its own DNA, but it's also, because the virus genetic material is a part of that now, it's also copied the virus. Now, that can happen over and over and over and over again, right? So you can end up with, pen's running out, thousands and thousands of these cells, and each one, wish I hadn't drawn that many, right? Each one will have its own DNA, but within its own DNA, it's also got the virus's genetic material attached in there. So that can happen over and over and over again. But at some point, what can happen is for one of these cells, it separates out. So it goes back to what it was at the start. So we've got the bacteria DNA by itself and the virus DNA by itself. And then when that happens, it can go back into this cycle. So this is called, this has got its own name, lysogenic cycle. So we've got virus injects itself, genetic material there, and then either copies the genetic material, assembles the protein coat, loads of viruses there, spits open, they can go and infect more, or the virus genetic material combines with the chromosomes in the bacteria. Bacteria does mitosis, making loads and loads of copies, so up here, and then at some point, so it can either go straight from there to there, or up and make loads to there, and at some point it can just keep going around in that circle, or it can go back to the lytic cycle, which leads to more and more viruses. getting produced. So this is how viruses can sort of lay dormant, right? So you can be infected with a virus, it can be doing the lysogenic cycle, you might not necessarily have know or being aware that you've got the disease, and then at some point it goes through the lytic cycle and starts damaging cells and you have the symptoms. Or maybe you have a virus, it's going through the lytic cycle, you're having symptoms, you're getting quite ill, and then you recover from it, but some of the virus is dormant in your body this way, and then it can start reproducing, and then if it comes back to here, you have symptoms. So key point, what is a virus? So a virus is genetic material covered in a protein coat, also called a capsid. Lytic cycle, and the key points of this for the exam, not just copying the genetic material, but producing the protein coat and assembling the protein coat. And the very last part of this video is you can measure the effects of viruses on a bacteria by using an Agar plate. So let's just draw a rubbishy picture here. Right. So Agar plate, you've got, it's supposed to be 3D, right? But then if we look at it from above, it looks like a circle. So you have a thin layer of jelly, you grow a load of bacteria on it, and then the bacteria, you can see it on the surface. And then if you infect it with the virus kills the bacteria, and you can see that because it makes little circles where, so it's like the virus was there and then it's spreading outwards, killing all the bacteria. Okay? And then how can you measure the effect on it? Well, the bigger the circle, the more bacteria have been killed. And just like in maths, you want to work out the cross-section of a cross-sectional area. So it's just the area of a circle. So it is pi r squared, where r is the radius. That's it.